So moving on to the fourth characteristic of the body plan, we are dealing now with the body cavities. So most triploplastic animals, they possess a body cavity. A true body cavity is called a coelom, and this is derived from the mesoderm. So let's add a little bit to the definition of a coelom or the body cavity. So this is a fluid or air-filled space between the digestive system and the body wall, and we human belong to these triploplastic animals, and we have such cavities. So we have, for example, the thoracic cavity, we have the spinal cavity. So we are among the triploplastic animals that we have a coelom. So the importance of having these cavities is that they provide protection of the vital organs. So as I have mentioned, the vital organs are protected with these cavities. And the second function of having this cavity is to allow for independent growth and movement of internal organs. So imagine that we said that it will be present between the digestive system and the body wall. So imagine the content of the digestive system is expanding. So if we have a body cavity, this will allow the movement of these internal organs. This will allow the growth of these internal organs independently. So one organ can expand without affecting the others or move. So it's a sign of development of these animals, and it is an advantage for the animals to have such a true coelom or a true cavity. So animals can be divided based on the presence of the coelom into the following types. So we have a coelomate. So as usual, this prefix means no or without. So these are the types of animals which don't have coelom. And we have coelomates. So these are animals with true coelom. And why we are saying true? Because we have another group which have what we call pseudoceromates. So pseudoceromates, they have a pseudocoelom, as we will show in the next slide. So the first group, and we are listing them here in terms of complexity or in terms of development degree. So the simplest type of animals are those who are characterized by having no body cavity. So these are a coelomate. So as you can see here, we have the ectoderm, which we have described, the outermost layer of the germ tissues. Then we have the endoderm, which is the innermost layer, and we have the mesoderm. And as you can see, there are no cavities. So this is the first group, the coelomate. So if you remember, the endoderm will surround the digestive system. So what is inside here is the digestive system of this flatworm. So there is no space for the digestive system to expand or to grow. Then the next group will be called the pseudoceremate. So pseudoceremate are characterized by body cavity that is lined by both the mesoderm and the endoderm. So let's see, and this follows this one in terms of development. So it is animals having pseudoceremate, they are more developed than those who are coelomate. So this is the ectoderm, the outermost layer, then the endoderm, the innermost layer, then the mesoderm. So the endoderm, again, here we have the gut, and here we have what is called as pseudocoelom. So if you focus on this white area, you'll find that this white area is actually in between two layers. This one shown in beige color and this one shown in pink. So the beige color is the mesoderm, the pink color is the endoderm. So that's why we say this body cavity is formed by or surrounded by both the endoderm from inside and the mesoderm from the outside. The third group, and again we are following the development line, so these are more developed animals. They are characterized by having a true coelom. They are called Celiumate, and now they have a true body cavity that is completely lined with the mesoderm. So it's completely surrounded with the mesoderm. So here we are showing the coelom. So if you focus on the cavity of the coelom, you'll find it surrounded from both sides with the mesoderm, and those having this type of coelom will be characterized as Celiumate. Then moving on to development, the next slides will focus on answering the following questions. How do the embryonic cells divide? How does the coelom form? How does the digestive tract develop? So these questions are important because they help us to categorize the animals as either protostome or deuterostome. So stome means mouth, remember it is similar to stomata when we talked about plants. So protostomes means, proto means first, so these are the animals in which the mouth will be formed first. Deuterostome, deutero means second, so these are the animals in which the mouth has formed second in terms of development. So this is the question that we are trying to answer in the following slides. How do the embryonic cells divide? How does the coelom form? And how does the digestive tract develop? You can say that this question means that did the mouth appear first in terms of development, or did the mouth appear second in terms of development? So let's start with cleavage, and as we said, cleavage is the rapid cell division. So starting from the 8 cell stage, this 8 cell stage will start to undergo continuous cell division. If number one, the arrangement of the 8 cell stage is spiral, so spiral means that the two tiers of the cells, each cell is a little bit rotated, it's a little bit angled in relative to the one below. So if it is spiral, and if the fate of the cell is determined, so each one of these 8 cells has a determined fate. If these two criteria are there, we will consider the animal to be of protostome development. In comparison, the deuterostome development is characterized by a 8 cell stage that shows radial arrangement. So if you pay attention, each cell is directly under the one, so each cell at the top is directly above the one underneath. There are no angle in this case, and the most important thing is that each one of these cells has indeterminate fate. So the fate of each one of the cells is indeterminate. So if you take any of these 8 cells, it can cause the development of a new organism. And this is actually the reason for the development of identical twins. So we, as humans, we belong to the deuterostome development. Some other animals, they are protostome development. So if you take one of the 8 cell stage, if we take one out, it will not result in the development of a full organism, or it will result in severe defect in the development of the embryo, because each one of these cells has a determined fate compared to the indeterminate fate in the deuterostome. Moving on to the celium formation. So we already covered the definition of a celium and what are true celiumates. So here we have two types of celium formation in the true celiumates. So the first type, where the solid masses of the mesoderm split. So as you can see, these two masses of the mesoderm shown in dark pink, they split, they come to the anterior end or the plastopore, and the cavity inside develop. So we will develop a celium inside these two masses of mesoderm. This type of development of the celium, we call it the development of protrosome. In comparison, in the deuterostome development, what is happening is that the celium folds from the archenteron. Remember that we said this archenteron is the digestive system of the animal, so the digestive tract, I mean. So this digestive tract will fold to the outside, creating the celium. So it is resulting from the folding of the archenteron. In this slide, we are going to discuss the fate of the plastopore. So let's first define what is plastopore. So the plastopore forms during gastrulation and connects the archenteron to the exterior of the gastrula. So if we go back to this slide, during the process of gastrulation, the invagination of the blastula, we have the pore, the opening of this blastula, we'll call it the blastopore. So this blastopore connects the archenteron, the digestive tract, to the outside or to the exterior of this gastrula. So we have two fates of this blastopore. This blastopore can develop into the mouth. So animals in which the mouth develops first from the plastopore, we will call them protostome. So protostome development is the development of the mouth from the plastopore, and as we said, proto means first, because the mouth will develop first, then the anus will develop second. Then we have another type of development called deuterostome. Deuterostome means the mouth came second in development. So here the anus will develop first from the plastopore, then the mouth will be the second in terms of development. And by this slide, we reach the end of this video about introduction to animal diversity. Hopefully it was useful, and thank you so much.